Yours.
Thank you, Roshan. It's a pleasure to be here. And as you indicated, am an employee with the American Bird Conservancy. I have worked in invasive species for a lot of years. I started with the Maui Invasive Species Committee back in 2002, worked with them as the manager for about 14 years, but I guess little fire ants and coqui frogs weren't challenging enough, so I accepted the opportunity to work on mosquitoes. And been working with a great team of people, a partnership that came together to try to address the problem of the imminent extinction of our forest birds. And just at the outset, I want to stress that we really are at a crisis crossroads for this, this situation with our forest birds, including the survival of at least a dozen species that are found nowhere else in the world. So I'm going to share a little bit about what we're doing and what our strategy is, and then hopefully entertain some questions. So when most people think about our forest birds, they think honey creepers. It's the word that comes to mind. And for me, even though I had a strong background in birds, when I think honey creepers, I think brightly colored, the decurved bill, nectar eaters. But it turns out that that's not the full story. And so what is so special about our honey creepers? And I know a lot of you know this story. It's certainly shared many times over the years. So there's just a couple of things that I want to highlight. We've had at least 56 different species of honey creepers since their time beginning in the Hawaiian Islands. Evolved from one or two rose finches species that probably got here through a process known as eruption, where greener pastures, they're looking for greener pastures, may travel in flocks of thousands, even 10,000 birds, and enough landed here to start colonies. And all that happened somewhere between 6 and 7 million years ago. And so our current species that we have here diverged from those original colonists somewhere between 2 and 6 million years ago. And given the isolation of the islands, as well as the closeness of them, they took advantage of the different niches, the different foods, the seeds, insects, nectars, and even a snail eater. And that's how we end up with this amazing diversity of not just bills. People focus on the bills, but a whole different suite of different species. And so what's the situation with them? They are in serious peril. Out of our 15 different species that are here in the main Hawaiian Islands, 12 of them are either vulnerable or threatened and endangered, with the biggest proportion of them in that threatened and endangered category. And only 3 of them are really considered fairly stable. Of those, our most imperiled honey creepers, we have some that have fewer than 2,000 individuals left. And just recently, the kiwikiu, the Maui parrotbill, has been estimated at somewhere around 150 birds. That's getting pretty bleak, really getting down there. And also just want to point out that it's not only our honey creepers that are at risk. We also have some other forest birds, endemic forest birds in the islands, including Elapaio, and then also several thrush species that are also at peril. And also want to point out that I was just thinking about this today, about how, again, our forest birds were here for millions of years, from the sea level up to the top of the forest. And when our first Polynesians, Hawaiians arrived in the islands, those birds were here to greet them with their beautiful plumage and their songs. And as a result, Hawaiian forest birds feature prominently in a number of Olelo Noia Hawaiian proverbs. And of course, we know that birds have been very important as a part of traditional practice, including lei and feather work. And so still very connected to our ancient and current native Kanaka Maoli. So a lot of different things that have caused the decline of our birds in the Hawaiian Islands. We're familiar with those different problems, habitat loss, invasive weeds, predators, cats, rats, etc. But the thing that is really driving our species to extinction now is avian malaria. And what is that? It's a intracellular parasite. And as we've mentioned, the birds have been evolved for millions of years. Mosquitoes only arrived in the Hawaiian Islands in the 1820s, the first one being Culex. And then the first documentation of Plasmodium relictum wasn't until about 100 years ago. So while some birds are starting to develop some immunity, most of them have not done that quickly enough. And the avian malaria is transmitted most readily in the lower elevation forests where it's warm and has previously been considered largely absent above 5,000 feet. Just quick fact, we have 6 biting mosquito species in Hawaii. 2 of those are night biters, the rest are day biters. So most likely, if you were being pestered in the nighttime by a mosquito, it's probably Culex, the one that is hammering our forest birds. So please feel free to go ahead and squish as many of them as you possibly can. Again, this is our culprit here, the southern house mosquito. And as I'm sure all of you know, we don't have any native mosquitoes here in Hawaii. Yeah, it can the females can complete the whole life cycle can be completed in 1 to 2 weeks. So from the time eggs are laid to adults emerging, something like 7 to 10 days. A female can lay about 5 egg rafts, which can have 200 to 300 eggs per raft, and do that within the course of her lifetime, maybe a month or 2. But that process depends on temperature and humidity to a certain extent. And also, of course, only the females bite, and they do need that blood meal in order to lay the eggs. So with the warming climate, both the mosquitoes and the Plasmodium have been pushing up into those higher elevations, the last refugia for our forest birds. And that is what's really causing some of our species, like the kiwikiu, to plummet. So those refugia are disappearing, and there have been some really heroic efforts to try to save some of our species, including a translocation project here on Maui for the kiwikiu. Moved a number of birds, both captive reared and wild ones that were captured to habitat that had been improved over a really long effort and big, major effort process. And within days of release, all of those birds were gone. And avian malaria was clearly the culprit. So what are our options? In 2016, after the World Conservation Congress held here in Hawaii, a group of experts from around the world gathered to consider what would a mosquito-free Hawaii not just look like, but how could we possibly get there? And one of the options that they homed in on, was the Wolbachia strategy. Wolbachia. So we've been talking about the mosquitoes. We've been talking about the protozoan, the relictum, Plasmodium relictum. And so here's another organism that we're focusing on, which is Wolbachia is an intracellular bacteria. It is found naturally occurring in the wild in an estimated 60% of all insect species. So it's ubiquitous, it's out there. It's also present in our Hawaii Culex mosquitoes. It's passed on by the female, and it maximizes its spread by manipulating the reproduction of their hosts. And that's a key feature that we are planning to take advantage of. And as you'll see in a little bit, it's not something that we just made up or came up with ourselves. And this came out of supported by that group of folks, but also is occurring worldwide for public health purposes. So the way it works, simply described, is you clear the Wolbachia from our Hawaii mosquitoes. And this has been going on in a lab in Michigan. So you basically use tetracycline to get rid of the bacteria that's there. You then reinfect them with a different strain of Wolbachia. So not the one that they naturally have, but a different one. In this case, the one they're using is the strain that's found in Aedes albopictus, or our Asian tiger mosquito. And when the males, and then you only have males mate with the wild females. So if a male that has that new strain of bacteria, the Wolbachia, mates with a female that has the different one that we already have here, no mosquitoes are produced. So they may lay eggs, but nothing is viable out of that. And so over time, if you flood the population with enough of these males with the different Wolbachia strain, and then the females go on, well, here's a male to mate with. Over time, the population is suppressed. It's reversible because this is not a gene drive approach. It's one that requires continued production and then release of these males with a different strain of bacteria. So as I said, this approach, this Wolbachia approach, has been used around the world. These are places where it's been used for public health purposes to suppress mosquito populations that carry things like dengue. And so we know that it works and it can be done. There are some significant differences between the kinds of strategies or places where they've done this and for health versus conservation purposes. Generally, much smaller scale, smaller urban district release. They drive down the streets with fans and roll down the windows and blow them out of a tube. And of course, everyone can support getting rid of mosquitoes. Well, almost everyone. I don't mean that like they do serve purposes in natural environments. On Hawaii, again, no native mosquitoes here. But so here we're talking about much, much larger scale, remote forest. We're going to have to use helicopters, probably drones, and we're talking about it for a conservation purpose. It is a truly innovative project that we're working on here at a huge landscape scale. Again, just if you've worked in conservation here, some of the terrain challenges that we're facing. So where are we? The transinfection has been done, as I mentioned, at a lab, Michigan State University. They've successfully infected the males with that different strain, and they've shown that when mated with the females, they don't produce viable offspring. So that is a big, big win. That's just happened in the last month or so. And then we need to get state, federal permits to go through that process because it will be considered as a biopesticide. So we'll be following pesticide labeling and registration requirements in order to use that here in Hawaii, as well as all normal public compliance, NEPA compliance kinds of requirements. Our overall steering committee has broken up into different subcommittees looking at operational issues, research, budget, community engagement. So really trying to be as focused and productive as we can be. This rather busy table produced by USGS just shows some of the research that's been done in the Hawaiian Islands. So we know a lot about avian malaria, we know a lot about our mosquitoes, but there are also some big gaps that we are working to fill so that we're sure we're releasing the mosquitoes where we need to release them and in the numbers that we need to release them. And then as well, the idea of using drones, people are looking into that elsewhere in the world to get into some of these inaccessible areas. Our community engagement challenges, some of our challenges is that people don't really know our forest birds. It's kind of easy to fall in love with monk seals and albatross that you can see in some places, but forest birds are hard to see to begin with, even if you're a talented birder, and our forests are not that accessible. And then additionally, it's kind of a complicated process to explain, and anytime you start talking about technology, sometimes red flags go up, or flags anyway, for folks, and then it also often gets lumped with, oh, you're talking GMO. This is not a gene drive approach. We're not changing the genes of the mosquitoes. But we also have to be careful not to explain that in a way that makes it sound like that's a bad thing, because lots of people are looking into that as an option. So we're in the midst of our community engagement process, identifying key stakeholders, having meetings, doing outreach of not events, but talking story individually and then with groups. and including our resource users, elected officials, and conservation organizations. And also, we will be connecting with people who we expect could be opposed to it. As I mentioned, it's a great group of people and organizations involved in it. When I started, there were 6 different agencies. We now have 10 that are active partners, plus our forest bird projects are involved. And I just really feel honored to work with all of the people who have dedicated their time and effort and resources to this project. We also have support from our agency directors at the highest level. So we're hopeful. We think it can be done, and we will need your support, and especially when it comes to compliance issues, but also just to help get the word out about what an important issue this is. So thank you. Happy to take any questions.
Thanks, Tia.
Yeah.
Questions for Tia? So Tia, I have a question. So Wolbachia is isolated from the mosquito, yeah?
It's cleared. Initially, the background Wolbachia is cleared from our Culex mosquitoes. And then yes, in the lab, and I don't know how they do that, but in the lab, they have to be taking, if that's what you mean, isolated, they have to be taking the Wolbachia from another mosquito and then injecting it into—
Feeding or injecting?
Injecting. They inject it.
Inject each mosquito?
No, unfortunately, that would be, that's what I thought, like, wow, that's a lot of really tiny
so
they inject it, and then they rear them. So they get that strain established, and then they rear them. And then what they have to do is do sex sorting, because you only want to release the males with this new strain. And so sex sorting, they have different ways of doing that. But yeah, it's really interesting process.
So I think there is one question in the chat box from Kylie. Can you?
Okay, so are they, although they're not producing more offspring, are they still able to transmit avian malaria? Are the females, can they still transmit it? Yes. No, they can still transmit, because remember, the females are the ones that are, it's transmitted. The avian malaria is transmitted when the mosquito bites the bird. And so yes, they can still transmit. So there are different approaches that have been made that have been used for public health purposes, and that folks are also looking at, which would be that disease blocking approach. It may be that if you do the super infection of using a couple different strains, that might result in blocking the transmission. One of the challenges with that approach is that then it becomes more like a disease, like you're managing the disease, or you're trying to block the disease as opposed to controlling a pest. And that could trigger something more akin to like Food and Drug Administration regulation, which would be more onerous. But yes, people are still looking at that, trying to figure out, are there also ways to stop the transmission?
Okay, so I have another question. So what I understood from your presentation is, yes, DOA has already gave permission to use Wolbachia, right?
Who did you ask has given permission to EPA?
The HDOA there is a permission involved in this use of Wolbachia, yeah, in a cage experiment, I guess.
Yeah, well, we don't have any permissions yet, but it has been for other species. The EPA has given that permission for other species, so there is a precedent for that, and we're working closely with the company, and it's actually University of Kentucky that developed the patent for this process. So we're working closely with them, hoping that we can take advantage, leverage some of their previous data and their work that they've done. So there is an existing precedent for doing it. And then we also have to have a permit at the state level to bring them back in. And University of Hawaii is involved in that process. They'll be the permit holder for that.
There is a couple of questions in the chat box. The first one, Sabrina. Is there
Yeah.
a way to get involved? And that's community involvement, yeah.
Yeah, right now it's as a member of the public, I think that we would love to hear from folks who want to be kept informed of this project, because there will be that public process part where we will be asking people to come and express your opinion about it. So if you want to, I'll put my email address in the chat if you want to reach out to me. And then the public reception to the technology, it's actually been pretty good in other places where they've done it. Of course, it's where they're getting rid of mosquitoes that are bothering people. But even some of the initial focus groups that have been done here in Hawaii, people are like, yeah, get rid of them. It's like they're not native and they're decimating our birds, and maybe they will even, maybe will even be some benefit for people. And then another question, would this only affect the population of Culex mosquitoes? Yes, because it's interfering with the breeding of that species. So no other mosquitoes at present. But Hawaii Department of Health is looking at it for some of our species that are problematic from a public health perspective. They're not as far down the path as we are, but there is interested in that for other species like Aedes aegypti. For public health purposes. Great questions.
Any more questions for Thea? We have still a couple of more minutes.
I have a quick question. I was just wondering, how exactly, I guess, does this, inhibit the reproduction between the mosquitoes so that it doesn't produce any viable offspring?
Yeah, it's called this incompatible insect technique, and it's also what's called cytoplasmic incompatibility. And it has to do with, I think of it as it has to do with the fact that it's the Wolbachia itself, it wants its own strain to keep going. And so if another, it makes it in the gametes don't make it, I'm not good at explaining that, but it's the Wolbachia itself has a number of techniques to stop reproduction, like male killing, sterilization, and then this cytoplasmic incompatibility. So it is a reproductive inhibitory process.
Okay, great. Thank you so much.
Thank you. And this will be the last question. Has this approach been used for several mosquito species? Yeah.
Aedes, yeah, that's a good question. I know it's been used for, I think, for Aedes aegypti and Aedes albopictus. But I can while we're going on to the next thing, I'll put that in the chat just so we're clear on that, because I don't want to mislead you.
Okay, thank you, Thea. That was very good information and very new technology in controlling insect species. Thank you.
Thank you, my pleasure.
And our next presentation is from JV Friday, updates and new information on rapid OHIA. And JV extension extension forester for CTAR, and he's based on Komohana Research and Extension Center, Hilo, Big Island. JV, it's yours now.
Okay, thank you, Rashan. Now I'm going to do the thing where I'm going to try to share. Are you seeing my?
Yes.
Are you seeing my presentation or
Yes.
the other mode?
Presentation mode now. Yes.
Okay, so you're seeing the right one.
Yeah. Do beetles carry the disease? Is that the first slide?
No, it isn't.
Doesn't look like a first slide though.
It's not. I guess I'm back in the middle. All right. Sorry, folks. We'll have to start over again. Okay, here we are.
Yes.
Here's the title. Here we are. So I figured what I would do was start out with where we have detections. And I'm assuming everybody knows we have two species of Ceratocystis, the pathogenic fungus that causes rapid OHIA death. And for Hawaii Island, basically we find both diseases across the entire landscape of the island. I don't know how many thousand detections we have, several thousand detections here. Kauai, as of September, and I'm sorry I haven't updated this since September, there are about a little over 150 detections of both species. And again, Leukohea is the more virulent one that's caused something like 85, 90% of the mortality in the state with this. Juliohea is probably been here longer and much less virulent. Now, why do we have both on Kauai and Hawaii Island, but not between? This we don't know. This remains a mystery. Oahu, there are only 5 detections, and they're all Juliohea. And let me emphasize that we detected those, we meaning the good folks on Oahu who are out there searching for this, because they were looking for it. 5 dead ohia trees on an island would not have gotten anybody's attention prior to having 1 million trees killed on Hawaii Island. And our estimate of Hawaii Island is there have been 1 million trees killed on a Hawaii Island. And then Maui had 1 tree. It was Juliohea. They cut it down, they burned it, they dug it out. So as far as we know, there are no positive detections on Maui. So that's currently the state of the distribution of the disease in the state. One of the things that this is actually old news, but I want to circle back to it, is one of the critical things is that ceratocystis is a wound pathogen. So the fungus needs a wound to get into a tree. This is an example. This is a tree in Ka'u, so it's on the rocky lava lands. Some animals, goats or sheep or cattle or something, have been peeling the bark off the trees. You can see the fungus there sporulating on the injury there, and this grove of trees died. So most of our forests do have a lot of feral animals on it, cattle, sheep, goats, and pigs. We see a lot of correlation between the number of animals and the disease. So this is actually right off the Hawaii Volcanoes National Park's website. These are the green and red circles are dead ohia trees. The red circles are the ones that were detected to have rapid ohia death. And then I drew in the fence line, because this is the Kahuku unit, and there's a fence line about halfway up the Kahuku unit that the sheep, the feral sheep and goats are below the fence line, not above the fence line. So what you see there is a lot of disease below the fence line, not much disease above the fence line there in the Kahuku unit. Now, this is a kind of complicated slide, but it's pretty interesting, and I want to step you through it. This, if you recognize the area, is Volcano Village. And the white lines are fences. The red and purple dots, sorry, the large red dots are trees that had symptoms of rapid ohia death and were sampled and tested positive for rapid ohia death. The green dots are trees that had symptoms but were tested and were not positive, so something else killed them. The small red and purple squares are trees that were symptomatic from the air. So the whole island, or at least all the ohia forest on the island, have been flown by Greg Asner's outfit, and they map symptomatic ohia trees from the air. And a subset of these are sampled, but of course they have, any one flight has maybe 50,000, and we don't go sample all of those. The areas that I showed, the Ola'a tract and the other little square there, were largely ungulate free. They're managed by part of Hawaii Volcanoes National Park. So they're inside the fence areas there and managed as ungulate free. The same forest on the other side of the fence is mostly state forest or private forest, and it has a lot of animals in it for hunting, mainly pigs in an area. So what you see on this is you see a lot of disease going right up to the fence line, and then some inside, but nowhere near as much inside. With the pigs, the earlier slide I showed you looked like it was sheep or goats peeling the bark off the tree. Pigs are tearing up roots as they root around. Now, we found out that it does not seem that trees take up the pathogen through their roots. And this is different than a lot of other pathogens. There was an experiment last year where Stephanie Yelnick at USGS planted seedlings underneath infected trees, And over the course of a year, none of the seedlings got the disease. Some of them were killed by pigs and whatnot, but they didn't die of rod. But once you injure the roots, then the fungus can get in. So we know these forests are full of pigs. We know the pigs root around and they tear up roots and they eat roots and everything else they dig around, mostly looking for earthworms. We know the fungus can get through the roots, and we know the fungus is in the area. And then we see this as a landscape pattern. So reinforcing this idea that it is animal damage to the trees that is allowing the trees to get the disease. Now, 2020 update, I don't have the photo of this, but the 2020 update, 2020, we saw a lot of additional mortality in Olaa tract. Not down in this little square here, and not above in this little square, these other fenced areas, but Olaa tract, a lot of mortality. When the teams went back in there, they found the fence had been breached and a number of things, and a lot of animals were in the fenced area. Both cattle and pigs had gotten inside that. So the change between when this photo was taken and the year later, the fence went down and animals got in it. So again, that's indicating that the animals have got something to do with it. Now, an injury can only get infected for a short amount of time until the wood dries out or whatever. So there's always reason to, and the park has been really diligent after seeing that. In fact, by now, maybe the animals are all gotten out. They got in there, they repaired the fence, and they're getting rid of the animals to keep protecting it. So protecting the forest is the best thing we can do, is the take home of it. You know, Roshan, I'm not watching the chat the way I've got it set up. So if there's chat that needs to be, otherwise I can take questions at the end. Is that good?
Yes.
Okay, got it. Okay, next project that's going on, and I just grabbed a few projects here that I thought people would be interested in. Next project that's going on are beetles carrying the disease. Now, a lot of pathogens are insect vectored, as we know. We see an association between rapid ohia death and beetles. Earlier work with Curtis Ewing, when he captured beetles coming out of bolts that were infected, about 2.5% of them tested positive. The beetles tested positive for ceratocystis. But are those beetles moving the disease to a new tree or not? So a recent study by Kyle Roy looked at, well, can a tree get the disease by being infected with a beetle? So what they did here is they took beetles and they marched them across a petri dish full of the fungus. I don't know how you do that, but entomologists do. They made it so the beetle has got fungus all over it. Then they took the beetle and they made a tiny little gel cap and put the beetle in and trapped it and glued it to the tree. So the only way the beetle could get out would be drill a hole in the tree. So an infected beetle drilling a hole in the tree, and then what they found here is the beetles did indeed infect and kill these trees with rapid ohia death. So we do know that beetles can infect. We do know that beetles can infect and injure the trees if they drill in. We don't think that's the major way that the disease moves around. We don't think it's mostly. We don't think that it's mostly transmitted by beetles. Okay, another project. Ryan Peroy here at UH Hilo has drones and does drone surveys to look at how the disease patterns go in the forest, in a forest stand. So drones are great because they're easy to fly. They get you nice pictures and they're really cheap. All it takes is a technician and a pickup truck and a drone, drive out, throw the thing in the air, and you got your images. The problem is they can only do a small area. So starting out with 400 or 1,000 acres, something like that. Whereas the airplane can do the entire island, but it costs $250,000 to get the airplane in the air versus the drone is pretty cheap. But one thing Orion has been able to do is there are several locations on the island where he has been able to do repeat surveys at a monthly basis. And so here's a place in Kau that had a few rod trees. And first we noticed it back in this was from 2010. So this was taken from some earlier imagery before Ryan got involved in this. So background before rod, about 1.5% mortality. And then what he was able to do is take a regular series of images and look at the mortality over time on this, I forget what this is, 200 acres or something this. I'm going to skip ahead in this. I will skip ahead if I can. Come on. by 2018, you had 25% mortality in the thing. So these are maps, and the interpretation of this is by hand. So a technician or a graduate student is parked in front of the computer, and they compare the images, and they can see from the image what trees are dying on it. So, but by looking at this, he's able to develop a mortality curve. So these are curves that are from 3 different sites on Hawaii Island. The initial numbers are from other remote sensing imagery, not Ryan's. And then once you see a lot of dots all together, that's Ryan's imagery on it. And the pattern we're seeing here is really interesting. For all 3 of these sites, it goes along at a low level of mortality, and then there's an exponential increase and then a leveling off. So you get this kind of sigmoidal pattern on things. One of the ideas of what we're seeing is there is an event that starts disease happening in a forest. It's not just a slow, steady progression. There's an event, then you get an explosion of disease, and then it tapers off. He actually has a newer graph than this. So this just goes 2019 to 2020. It doesn't flatten though. It keeps on going. So what you might have going here is an event, and we're talking something like a storm that will blow infected frass from another forest, break a lot of branches, knock trees over, whatever, and then you start getting disease locally coming from that. And then over time, there might be secondary spread, but it's really, the secondary spread isn't that severe. So it's just that low level, but it's these events over time, the events that occur that seem to kick off patches of mortality. And kind of that would explain the island-wide mortality we see here, because we'll see a forest that's really infected, and then a few miles down the road, it looks pretty good again. And so it's not just this moving evenly through the whole forest, but there are events here and there that are going on. Flint Hughes has about 250 plots around the island. A lot of data coming out of these, and some of them are 4 to 5 years old by now. Some of them are new. But one of the interesting tidbits of data that's coming out is trees less than 8 inches in diameter are much less apt to get the disease than larger trees. So they only, overall, over his whole data set, only about 5% mortality over the time he's been monitoring it of these small trees, like the one in the blue ribbon there. But the larger trees that are above 8 inches diameter have 13% mortality, even if you don't have a guy chopping into it. That tree was dead already. That's why that guy was chopping into it. But smaller trees have less mortality, and we think, again, it's that you have windstorms that break branches, that damage trees, that you get a lot of mortality, whereas these small, flexible trees, you have a lot less mortality on them. A less encouraging result is that he saw very few seedlings regenerated below 3,000 feet in elevation. And really probably what you're seeing is above, so the Ohia forest can be resilient. Even up to like the 1980s, when they clear-cut Ohia forests in Puna, they came back as Ohia forest. It was resilient. Above 3,000 feet, you're seeing Ohia regeneration in these plots. But below 3,000 feet, the Ohia regeneration is pretty much crowded out by all the weeds. So in these forests, they are not resilient. When they're hit by rod and hit by invasive species, you lose the forest. So like this forest here in Puna, the understory is mostly all weeds now. It's pretty much gone as a native forest. Okay, can we treat wood? One of the concerns, I know there's a concern for Maui people, there is a trade in Ohia logs. So balustrades, posts, architectural posts, that's the main thing it's trading. And these logs are shipped off the Big Island to other islands, and currently they are inspected individually. Which is expensive. Department of Agriculture costs them a lot to do it. It costs our lab a lot to do it. And no inspection is perfect. They could always miss something with an inspection. So can you treat the wood? Well, we did several trials on different ways to treat the wood. Here, these are posts like you'd use for a balustrade, run through a kiln, commercial kiln in Hilo. It's a 23-day drying cycle. The magic number is 140 degrees F, because we know that that heat kills the fungus. So for these small logs, run through the kiln, it killed all the fungus. So this was a trial with infected trees run through the kiln. This treatment was effective. It killed all the fungus in that. Department of Ag is accepting that as a treatment. And when those posts are treated, then they can be shipped inter-island. Now, what about big logs? So these are some large logs. So these were actually harvested from a forest where they had and were confirmed to have rod. This is a technology that these two guys from Virginia State brought out, Mark White and Xinjing Chang, brought them over from Virginia State. And this is something they have that they use there to disinfect logs for shipment to Europe, American trees, which have diseases they don't want in Europe. So this is a vacuum steam kiln, and it was a pretty cool process where they put the logs in and then they sucked in all the air out and created a vacuum and then injected steam. And the fact that it was a vacuum allowed the hot steam to penetrate the log really quickly. So these big logs, if you just had to heat them in a regular kiln, they'd be there for months and you'd be paying enormous electrical bills. But the vacuum steam heated it overnight to 140 degrees F. And that was 100% effective in killing all the ceratocystis in the logs. So, and again, when I say it was 100% effective, after they did this treatment, they took those logs apart and took hundreds of samples out of each log and tried to grow the ceratocystis from the samples, and they didn't find anything. So that's effective, that's effective. Now, the problem with the kilns, though, is they're expensive. That's a couple $100,000 investment in a kiln. What about just a fungicide soaking with boron or quaternary ammonia? These are things that are used to control fungus in wood. So Mark Hughes and other colleagues this is just new, reported this last month. He did a set of trials where he soaked infected wood in these two moldicide treatments. These are common available commercial moldicide treatments. One's boron and ammonia. And they found as long as you had a reasonable concentration, it was 100% effective. In these little discs, it was 100% effective in killing any of the fungus in the discs. However, they moved on to logs. Okay, what we really want to do is treat logs. Say at a number of logs, 1.5 inches to 7 inches in diameter, they soaked them in the fungicide overnight and they left them for 10 weeks to diffuse, which I guess was the regular procedure for that, and then took samples from the interior and the exterior of the log. The problem is that the fungicide only penetrates about 3/4 of an inch, and they sealed off the end, so this is radial penetration here. So the problem is the fungicide doesn't soak very well into ohia logs. Now, I remember Bill Cowan on Kauai was treating albizia. The chemicals soak right through the albizia log in nothing. And I mean, it's a great way to treat them. Ohia is hard and it's not penetrating very well. So when they looked at it, they did find living ceratocystis spores after the treatment. It was about 90% effective, but that's not good enough for an inter-island quarantine. So Department of Ag wouldn't accept that. So fungicides not really working really well. Can we protect live trees? This is actually a photo of an elm tree in Hanover, New Hampshire. When I visited this tree, I talked with a college arborist. This tree gets fungicide injections every other year, and it gets weekly inspections for symptoms of disease. And if they find disease, they give it more fungicide. Could we do something like this for exceptional trees? Well, in the lab, propiconazole works well to kill the rapid ohia death fungicide. And we did a few trials with pumping trees full of fungicides, then inoculating them with a pathogen. There was a high pressure, low volume, and a low pressure, high volume trial. And unfortunately, the results are mixed. Now, this really gets back to the disease triangle where you have a pathogen, a host, and an environment. These were done at 2 sites. At the high elevation site, which was cool, at the high elevation site was cool. A lot of trees still survived after quite a bit of time. But at the low elevation site, even though you protected a lot of the trees with the fungicide, most of them died, and all the ones that were inoculated otherwise died. So the inoculation with the protection with a fungicide, we're not really sure if it does more harm than good. So it's not something that we can recommend now. If you see so in conclusion, if you see a suspect rod tree, let the appropriate invasive species committee or the DOFA branch know. If it's on state forest, let DOFA know. Let the invasive species committee know if it's on private land. That's for Kauai, Oahu, Maui, Molokai, Lanai. Hawaii Island, we're only tracking trees in Kohala and Hamakua. The rest of the island, we know it's there. But let them know, and either DOFA or the Invasive Species Committee will take care of it. One of the things, again, we want to stress is because the younger and smaller trees don't seem to get the infection, and if you don't injure your tree, keep planting it. We do want to say keep planting ohia. We want to say keep planting ohia, keep using it to restore and grow the forest. And really the best thing to do is to protect the forests, in the forest from the animals that are damaging the trees. This is J.C. Watson's Ohia Legacy Project, and I think they have these annual ohia giveaways, and I think these guys ran out in half an hour. So there really is a desire for more ohia among the public. We do have a new strategic response plan. So if you go to our website, rapidohiadeath.org, you could download the whole plan. And I just gave you a few highlights of research. We have a lot of other projects that are going on that I didn't really delve into today. Most of our forests are healthy, even here still on the Big Island. Most of our forests are healthy, so we want to keep the forests healthy. And thank our funders. We've had a lot of different funders for this. Coming around, it's going to be a tight year. We've had a lot of funding in the past couple of years from the state, from DLNR, and that's going to be tight. So, so yeah, we appreciate the other things such as the Hawaii Community Foundation, Hawaii Tourism Authority. That's our website. We're also on Facebook and Instagram. I'm JB Friday. There's my contact information, and my new assistant is Hoku Pihana, who's also working out of here at Komohana. So with that, I'm going to keep this on and maybe answer a couple of the questions, and feel free to type other questions. What about rod on Kauai? Why is it on Kauai? No one knows. It's really very low levels. And again, you may think that 150 trees infected on the island is big, but we have north of 4,000 on Hawaii Island. But nobody knows why Luku ohia is only on Hawaii Island and Kauai. It is possible that it's possible that it moved from one island to another, but it's also possible that wherever it came from, the same pathway it came to Kauai and the Big Island. So maybe it was on an imported plant that someone imported to Kauai and the Big Island at the same time. So we don't really know that. Some of the genetics, and this is research I didn't get into, Tom Harrington at Iowa State is doing some of the genetics. He's placing Luku ohia on the Big Island, has probably been there more than a decade. And again, at first, it doesn't look like anything out of the ordinary, so you don't notice it. Let's see. The areas above the fenced Ola forest have a lot of rot affected trees. It tends to drop off as you go Mauka in that area. So above the Ola forest, I'm pretty sure mostly that is Pu'umaka'ala. And it would differ in Pu'umaka'ala because they're fenced and non-fenced areas there. So it's a pretty complicated pattern on the landscape. Could rod spread from ohia nurseries stock unaffected trees at a planting site? That's a good question. We don't think it's really, we think if seedlings are infected, they're going to die. I mean, it's possible that a seedling could have gotten infected the week before it went out. We think that's a pretty low likelihood of moving stuff around, of moving the pathogen around. So it could theoretically do that. I do know that you cannot ship ohia seedlings off of Hawaii Island. So nurseries here are not going to grow seedlings and ship them to another island, though, because that does fall under the HDOA quarantine.
Thanks, JB. We still have time for questions.
Hi there. I had a quick question.
Sure.
I was just wondering if there was any type of notable increase in cases on either island, correlating with maybe any increase in a specific type of invasive species, or if that maybe was something that doesn't really affect it as much as overall, like the overall impact of the invasive species.
Yeah, that's tough to say. I mean, other things that could have happened is likely the pathogen came here on a plant. Likely it was, and it possibly hybridized and mutated once it got here and somehow jumped to ohia. So, and the plant would have had to be out in the forest, right? If a plant stayed in someone's garden, maybe not, or maybe because the line between gardens and forests in Pune is not distinct, right? So people were planting stuff out, maybe it spread to that. But the moving it around, now, again, we're still really working on what the role of insects are, but one of the roles of insects is for sure drilling into trees, creating a lot of frass that blows around. So the beetles that are producing this frass are almost all, not all, but almost all invasive. They're ambrosia beetles like coffee berry borer and black twig borer. So they are also creating a lot of frass that blows around. So that's one invasive species. Plant species, the effect of the plant species is when the weeds come in, the invasive plants come in, they take up all the growing space. And the seedlings can't regenerate.
Okay, thank you so much.
JV, is that possible if the ceratocystis disease from other plants like sweet potato and taro might have mutated and infected Ohia on Kauai or Kauai Island?
Yes, we think that the ceratocystis came here, either was an undiscovered species that came here, which is possible because there's a lot of pathogens that we don't know, or it mutated or it hybridized. The Huliohea is in the same branch, the same clade of the family as the taro pathogen on Kauai. So speculatively, that could have mutated to Huliohea. Leukohea is not at all related. So it is related to some other pathogens that are pretty severe pathogens in Latin America, but not at all related. And neither is related at all within the big spread of ceratocystis to the sweet potato pathogen. But could something have hybridized with something else and some weird crossing that happened once and then it adapted to Ohia? Yeah, something like that could have happened.
Thank you. We can have one more question to JV. If not, we thank JV for a very interesting presentation. And before we take a break, welcome back again. So this is our second session, and this second session consists of 4 presentations. And out of those 4 presentations, we have a first presentation on updates on spread of coconut rhinoceros beetle and the response efforts from Kylie Kosaka and Koki Hutchinson. Is that right? Koki will be presenting this talk, and both Koki and Kylie is the outreach specialist for CRB, Coconut Rhinoceros Beetle Response Team. Koki, I will stop sharing this presentation. Okay, yeah, she is already there.
Okay, thanks.
So whenever you are ready.
Perfect, thank you. So thanks for joining us, everyone. My name's Kylie, and I'll be presenting with Koki. We're both the outreach specialists for the CRB response. CRB stands for coconut rhinoceros beetle. So it's this large black beetle, not native to Hawaii. The scientific name is Oryctes rhinoceros. And these beetles can get to about 2 to 2.5 inches in length. They have a prominent horn and strong forearms that they use to feed on the spear of the coconut tree and other palms. So what does the life cycle look like? Similar to a butterfly, coconut rhinoceros beetle will start their life as eggs, move through 3 larval instars, pupate, and emerge as adults. So they can fly as well. We've observed that they can fly up to 2 miles at a time, but they really do spend most of their life cycle in decaying plant material. So that could be mulch, compost, anything that's decomposing. And the average female can lay over 90 eggs in her lifetime, which is really incredible to think about the damage that one beetle can do. What does the current status and spread of this beetle look like? So This graph shows all of the trap detections that we've observed over the entire course of our program. Coconut rhinoceros beetle was first detected in December of 2013, and the response started shortly thereafter. One of the first things that the response did was put out all of these traps that you've probably seen if you're on Oahu Island. So that photo in the top right corner is a picture of our black panel traps. And the beetles are attracted to a pheromone lure. They fly into the trap and then hopefully fall into the cup at the bottom. So the traps across the island are our main way to detect where the population is and where we should focus our efforts. An important takeaway to take of looking at this graph is that we are seeing an increase in the population and increase in trap catches overall, especially over the last few months. Another way to visualize this is through this map. So this shows where our most recent detections were over the last two weeks ago now. Some areas to point out, we recently found one of our first detections in Pupukea. So that's very alarming to us. Thankfully, it is, after deploying more traps out there, we haven't noticed any additional catches. Some other places that are of concern to us is the spread that we're noticing out into the west side of the island. The red border indicates our buffer zone, so that's all of the area that we classify as concerning for coconut rhinoceros beetle. And another map showing where the problem is mostly is this one right here. So this is a heat map that indicates where our trap catches are concentrated across the island. So yellow indicates really high trap catches, whereas blue are smaller trap catches. Our main areas of focus right now include Pearl City Peninsula, so that's kind of that middle yellow and red dot, Iroquois Point just below that. And then we're really concerned about spread out into Eva Beach, around Waipio Peninsula into Kunia and Mililani. You'll notice that there are a lot of smaller dots out onto the west side, and we believe those to be human vectored mostly. We are kind of seeing an increasing trend out there, so it is concerning for us. But we're hopeful that the drier conditions out there will help to deter breeding populations from establishing. So where are our priority zones or our main areas of focus? Those areas that I listed earlier. But this graphic shows what that translates into as percentage of trap detections and square mileage. So Pearl City Peninsula, for instance, is one of our highest catch areas. Within the last year, that was the total amount of catches was 44.13% of our total catches overall. And it's the smallest area that we're focused on. So thankfully the problem is mainly in that area and hasn't spread to a larger area beyond that. But we are seeing spread somewhat into the west side and out into Mililani and central Oahu as well.
Thanks, Kylie. So normally at this point, we would give a kind of more in-depth look into what coconut rhinoceros beetle damage looks like. But for this group in particular, we wanted to focus it on some updates, assuming that you have some familiarity with coconut rhinoceros beetle and the damage that they cause. If you're not familiar, you can go to our website, crbhawaii.org, which will also be at the end of these slides. But something that we really wanted to touch on today was the risk of coconut rhinoceros beetle to our native lolu palms. So unfortunately, the CRB response has observed that more of the trees that we found to die from CRB damage have been native lolu rather than the coconut palms that we might expect. And these tree deaths have occurred at LCC's campus. So in trying to address that issue and protect lolu palms, our field operations team is working with some partners to test the efficacy of traps that are in more remote less accessible locations. So to see if those traps could be effective, even if they're only checked every 3 months or so, to protect some of our more heavily Lolu populated forests. But definitely this is something of concern. We can't say that CRB prefer Lolu at this point, but just that we have seen significant damage to these, and instances of tree death have been, more instances of tree death have been found with Lolus than coconut palms on Oahu. And then next slide, please, thanks. So going into some more field updates, we'll cover about our canine team, some results and what we've been doing with the tree injections, and then looking into our next year, how we're going to do more contracted green waste removal. So with our canine program, if you haven't already seen our social media, we love to share about our canines. So we have a team right now of 2 canines and 2 handlers. So the dogs are trained to smell larvae in potential breeding sites and then have a sitting response. And so that alerts their handlers and the rest of the team that it could be a CRB positive site. And so the canines have successfully detected breeding sites and they're working to hone their skills even more. Since it's a new program, they're working out some of the logistics with how much you're supposed to trust a dog if you're not able to have the field crew verify right away that it is a CRB site. But canines hopefully will help with allowing our field crews to really focus on those positive sites. And then next slide, please. Going into some of our treatments, so imidacloprid has shown good results at being fatal to CRB. So 2 options for that. One is a granular pesticide, which is used at certain breeding sites where the mulch or breeding material can be spread thinly because it is labeled for lawn use. And then another option is to do tree injections, and we've only done these in a few select areas that have been frequently landscaped in our really hot spot areas. So trying to prevent the risk to potential fruits. And then with the outreach team, so Kylie joined the CRB response in July and I joined in August. So we're really working on reaching out to some partners, continuing those relationships and establishing an online presence. So like I mentioned before, you can find us online at crbhawaii.org. And check out our social media. We'd be more than happy to connect. And we're glad to join this group because I think it's the first time for me seeing many of these faces. So that brings us to close to our end. Thank you again for having us, and please reach out if you have questions. I see some in the chat, but yeah, we can wait until the end or address them in the chat.
Thank you, Kylie and Cookie. So we will have back-to-back presentation, and we will have around 15 minutes of question answer at the end of our presentation. So next presentation is on managing invasive rose ring parakeets on Kauai by Jane Anderson. Jane Anderson is a research professor at Wildlife Research Institute at Texas A&M, and she's with us here in Kauai working with the USDA. Jane, it's the floor is yours.
Hi there. Let's see, is this on the right screen for my presentation?
I said yes.
Okay, yeah, okay. Well, thank you so much for the opportunity to present. As Rosan said, my name is Jane Anderson, and I'm with Texas A&M Kingsville. And today I'll be talking to you about ongoing work with rose ring parakeets on Kauai. The rose ring parakeets are a medium-sized parrot. They're about 40 centimeters long. They're native to equatorial Africa and the Indian subcontinent. They are very popular in the pet trade, and as a result, they've become one of the most invasive bird species in the world, as they've been moved around by folks and unintentionally released in lots of places. One estimate suggests their breeding range has increased by over 4,000 times since the 1960s, so they're rapidly spreading and fairly recent invasive species in the world. To date, there are populations on 3 Hawaiian islands. The first was introduced on Oahu in the 1930s, and an estimate from a couple years ago suggested there are about 4,700 birds. We don't know much about what's going on the Big Island. We know the introduction was fairly recent, but we don't really know how many birds are there at the moment. My work is on Kauai. We believe the population came from just a couple birds that were intentionally released in the 1960s. We are working on a genetics analysis to confirm that. The population remained pretty small for the first few decades, which is pretty common with this species, and was estimated about 2,000 individuals in 2011, 6,800 in 2018, and my team and I estimated over 10,500 in January of this year. And one of the primary issues we see in Kauai is crop damage. There are a lot of problems of parakeets taking corn from the large corn operations on the western side of the island, as well as seeing lots of crop depredation in the small fruit farms throughout the island. So everything from mango, lychee, longan, papaya, basically any sweet, fleshy fruit is getting hit pretty hard by these guys. Now at night, the parakeets congregate in roosts. So these big, big congregations of birds that sleep together. These numbers were from January of this year when there were 3 known roosts. And the numbers in the bottom indicate how many animals we estimated were at each roost. So about 6,500 in the Poipu roost, about 2,500 in Lahui, and about 1,500 in Kapa'a. What's interesting and important to note about these is these are all in pretty developed areas. Mostly the birds are sleeping in tall royal palms, which are mostly planted in residential and commercial areas, largely in parking lots. Interestingly enough, and notably, the Poipu roost is largely around tourist resorts and all on private property. So we see a few issues with these roosts, and besides the fact it's very loud to have thousands of birds together, there's a lot of poop. You can imagine 6,500 birds pooping in a pretty small area. So we know there's potentials for disease transmission. The odor is and cannot be overstated. It's pretty intense. Beyond that, there are seeds in the droppings, which is increasing rodents in the area. And we have a lot of folks reporting property damage. So today I'll be talking to you about a couple of tests we're working on for populations to reduce the population, strategies to reduce the population size. First, looking at feeders, and second, lethal control. So this is a feeder that was designed by the USDA, and you can see here it's been successfully used with another invasive species, monk parakeets. The idea with the feeder is that if we can use it successfully, it could be used with chemicals such as a toxicant or a contraceptive. However, we want to make absolutely certain that a native bird cannot access these things. So parrots have what's called zygodactyl feet. They have 2 toes pointing forward and 2 pointing back. And that's what allows them to climb up and down vertical surfaces and also hang upside down. So this trap is so clever because it's cheap, and it also uses this wire curtain around the feed, requiring the birds to climb and hang upside down to get to the feed. So in other words, birds that don't have the capacity to do that shouldn't be able to access the feed. However, before we can test this out in Kauai and see if it would work with the rose-ringed parakeets, we have to first just get them habituated to coming to feeders. What's really interesting is they use feeders throughout their introduced range, including on Oahu, but we don't know if parakeets using feeders on Kauai. So we began in January of this year with these platform feeders. Again, these have been successfully used with other invasive parrot species. They're just these big open containers out in the field, baited with commercial bird seed, and you can see they've got artificial fruits and decoy parrots. We left these out for 12 weeks and did not get a nibble from parakeets. We got basically every other non-native bird on Kauai, but no parrots. So we were wondering if perhaps these are too low. Again, they've been successful in other places with other species, but maybe the rose rings in Kauai don't like them. So we're currently testing out a couple other feeders. On the left, you'll see a cylinder feeder, again, just baited with commercial bird seed. And on the right, there's a feeder with whole peanuts. Rose-ring parakeets are said to love whole peanuts. Lots of folks use those to trap them. These feeders are elevated to about 10 feet high, and you'll see we also have decoy papayas and parakeets. We also have an MP3 player emitting rose-ring parakeet calls. Unfortunately, we have not seen parakeet use so far. They've been in the field for about 5 weeks. And we'll be running this through the end of January. So fingers crossed, maybe we'll see parakeet response to these. The other effort I would like to share today is roost calling. So a lot of folks think of roost calling as perhaps a silver bullet to control these guys, well, no pun intended. But you have all the parakeets together in one area, why not just shoot them there? So historically, this has been avoided in Kauai for two reasons. First is when roosts are disturbed, the parakeets will abandon the site. This is problematic for a couple reasons. First, we depend on the roost to estimate the population size. So if we lose track of the roost, we're going to have a hard time knowing what's going on with the population. Beyond that, as I mentioned before, the roosts are in very populated areas, and we are concerned that folks, especially tourists, won't particularly love folks out in the middle of the night shooting these beautiful green birds. Folks tend to be less opinionated about killing things like mosquitoes than pretty parrots. However, the COVID pandemic provided a pretty optimal opportunity to test out at least the second concern. Not only were there no tourists on the island, but also for a period there was a 9 p.m. curfew on Kauai. So the streets were pretty empty to give this a shot. So roost calling was funded by the county, and it was conducted by a private wildlife control company. And these folks were kind enough to allow us to collaborate with them, collect data, and actually turn this into research, which was just, I can't speak highly enough of them. It was really gracious. So they collected, they conducted calling for about 19 nights between March and June. They used a 25 caliber air gun. Effectively what this means is they were shooting individual birds one at a time. So we saw a variation in success in between the two roosts where they were working. In the Lihue roost, they were able to kill about 500 birds out of the initial population size of 2,500 over 3 nights. However, after 3 nights of effort, the birds abandoned the roost. So at this point, we don't exactly know where those remaining 2,000 birds went. However, the Puipu roost, the company was able to remove about 4,700 of the 6,500 birds at that roost site, and it appears most of the birds remain there. So we're collecting some more data. We're doing some work to try to figure out what the discrepancy was between those two roost sites and why it appears birds were more tolerant of this at one site than the other. The company saved over 3,000 of these birds for us so we could do some continued work, and we processed about over 3,000 birds. You can determine the adult males because they have dark rings around their necks, but the adult females are indistinguishable from juveniles in the hand. So we conducted necropsies to determine the age and sex ratio of the birds taken. Ultimately, with vertebrate pest management, it's most effective to control the adult females because they can move forward and create the babies. It's not as effective to remove juveniles or adult males. So I just put these together. This is brand new information. So these are the rough breakdown of the animals that were collected from the roost calling. You'll see the adult females were kind of low, or pretty low. We think this is likely because the calling happened during the nesting season. So the females don't come back to the roost while they're nesting, which makes them not accessible for shooting at that point. However, unfortunately, we don't know the exact nesting season on Kauai. So we have a lot more work to do analyzing these data, but ultimately, there we go. We'll be using these in our population models to project the future population size of the rose-ring parakeets, both as a result of this one roost call and as far as whether this effort is done again in the future. We are continuing to do roost counts, again, trying to get an idea of the abundance and distribution of parakeets on Kauai. In January, we are planning to do a big community outreach effort and ask folks to report when they find roosts in their neighborhoods. And there's a good chance I'll reach out to this group and ask for help spreading the word on that. So thank you in advance if y'all would help me reach out to folks with that request. We need a lot more information about these birds. Managing a species is contingent on understanding just the basic natural history. There's a lot of things we don't know yet. So there's a lot of work to be done, but ultimately we do recommend that the other islands that have these birds and potential future introductions, do keep a close eye on them and intervene quickly if possible. There's a lot of amazing folks helping with this project, but I think I'm running short on time. So my email address is at the bottom, and I'll be here for questions at the end of the session.
Thank you, Jen. So yeah, moving along, we have another presentation on Myopodium thrips, managing Myopodium's most menacing pests, and considering for thrips, early detection and rapid response efforts. From Nate Dube. Nate Dube is the manager of Oahu Invasive Species Committee. Nate, whenever you are ready.
I am.
Right.
Well, my kako, everybody, can you hear me okay?
Yes.
All right, so I'm here to talk about Myopodium thrips today. We'll first start out with a little bit of background. I'm sure most of the folks in this mini conference are familiar with our native Myopodium species, Nile. The main species is distributed across most of the Hawaiian Islands. It's Myopodium sandwichense. It's really dominant in the coastal strand, but it can grow up to about 3,000 feet. You'll find it in the forest and most of the major Hawaiian Islands as well. It grows across a variety of habitats and ecosystems. It also has the ability to grow upright and more woody, as well as more prostrate. The prostrate version we call Niopapa. It has many Hawaiian historical uses. You can see it was very important in water-based activities, which makes sense as it was a dominant coastal plant at that point. And here's a few pictures just so you folks can kind of see the differences on the left here. We have some upright Nio. Again, it's a pretty common plant. You've probably seen it in both landscaping and wildland environments. And on the right, we have Niopapa with a little zoom in to show you the more creeping ground cover growth habit. On Oahu specifically, we do have an endemic Myopodium species. So when thrips was detected, and I'll go over the timeline in just a minute, it was really important for us as an island to come together and pool our resources to do everything that we could to protect not only the more common Myopodium sandwichensis, but also the stellatum species. Stellatum was historically primarily found in southwest Oahu and in the Waianae Mountains. It was once very dominant. The current wild population, the numbers are a little bit unknown. They think it's about 150 individuals, but it is bordering right on the threatened label in terms of species designations by the IUCN. So it does prefer dry regions, and it makes sense why it would grow over in that region of Oahu. It's primarily found only up to about 150-foot elevation, so it's really more of a coastal plant. And it has shown a little bit more resistance to thrips infestation than the sandwichense. So those are the plants that we're going to be talking about. And now we're going to go to their most menacing pest, which is the thrips. Just a couple quick notes. You'll hear me say a thrips. Thrips is both plural and singular. So it was hard for me to get used to, but I promise that's the correct pronunciation. So they're these very tiny winged insects. And for the most part, they are huge pests and they damage quite a few commercial crops. They also have an incredible diversity. And the reason why that is important is because, as you'll see later as I go throughout this presentation, we've had a very difficult time finding effective chemical treatment methods. So the other interesting thing about this species of thrips, specifically the Myopodium thrips, is it's relatively new to science. It was first detected in California in 2005. Before that, it was only previously known to be native to Tasmania and found nowhere else in the world. So this was the first detection outside of Tasmania. 2007, they completed a species description. And then in 2008, some land managers and landscapers observed thrips on our native Myoporum on the east side of the island. So it was reported to the Hawaii Department of Agriculture. HDOA responded by initiating some delimiting surveys and conducting some assessments of certain infestations. And then as it was determined that thrips were really only present on Big Island at that time, the HDOA did suspend movement of Nio plants in our island. So fast forward approximately 10 years later, we first detected Oahu thrips on November 23rd, 2018. So since then, I apologize, this map isn't 100% up to date, and we'll get into that a little bit more too later in the presentation about funding. But basically, this is the current thrips distribution on Oahu. Within about a year and a half, thrips spread an incredible amount. I remember hearing one land manager on the Big Island talk about how he associated the spread of this particular pest to wildfire because of its capability and wind dispersal and its capability to really spread quickly throughout the islands. So since we published this map back in February, there have been a few additional positive detections. There are probably more detections that we're unaware of at the moment, but right now we have the Mauna Lua fish ponds at Kuli'o'o, so that's right down in the southeastern corner of Oahu, not too far from the urban core. We've also seen some wildland positive detections, which is really unfortunate because that was essentially the breaking point for when we decided that the infestation was a little bit beyond our control. So Kaluakawila, there were some wildland detections, and also since then there's been some at the Nike site at Pahole. There's another wildland detection, from what I understand, just south of that Kaluakawila point indicated in the northwestern portion of the island. And then there have been several other sites around the urban core. So because of funding, OISC has really stopped the coordinated effort towards eradication of thrips. At this point, it's transitioned into more of a management species. So what that means for us right now is it's up to all the different land managers, whether that be big land managers like the Honolulu City and County and Board of Water Supply, or the state DLNR, or even, you know, folks who just live in neighborhoods and might have a plant or two at their neighborhood. So if it's just a resident, we do try to go out and remove the plant for them if it's infested at their request. But other than that, we're really hands off at this point for this species. So I just quickly want to go through some damage identification, since I know a lot of you folks are not on Oahu. This species is very prevalent on the Big Island and now prevalent on Oahu as well. But I don't know of any other detections on the neighbor islands. So one of the key components to a successful response to this species, I think, will be catching it very, very early because of the speed with which it spreads across the landscape. So we'll just go over a couple of pictures to show you some damage identification. It's really important for folks who are going to be looking for this species to look for the damage and not the insect. Again, you'll see some pictures a little later on to show you the scale and just how tiny these species are. This is a great picture from Cynthia King that shows kind of over time what thrips do. On the left, you have the uninfested piece of vegetation. In the middle, it's early infestation. And then Once it gets heavily infested, the leaves start to deform with all the galling insects. Again, here's infested on the left, healthy on the right. Here, it's a little bit hard to see. It's hard to get a picture of Nyopapa damage, but this is what it looks like in a patch of Nyopapa. And essentially, these are all the different kinds of leaf distortion and galling that you'll see. It's very, very indicative of thrips. There are not other pests that we have seen on Nio plants that cause the same type of damage. Again, leaf distortion and galling until you get the end result, which is defoliation and typically death with these species. So one of the major things that we found when responding to this species was just how critical decontamination was. These insects are very tiny, they're wind dispersed. You might want to think about canceling operations on a windy day if you are responding to a thrips outbreak. Because of their wide diversity, they are also really good at developing chemical resistance. So that is, we'll talk in just a minute about some of the chemicals we've tried, but that is a big consideration. So you can't rely on a complete kill. Essentially, what you folks need to do if you are dealing with thrips is be very, very cognizant of decontamination. Try to use species-specific clothing, Tyvek suits when you can, bring lots of brushes, and allocate a substantial amount of time, I'd say, at least 30 to 60 minutes for inspections of each other, inspections of gear, and time to decontaminate yourselves on site so you're not becoming a vector of spread for this species. And if you don't have any considerations for decontamination, if you're just going gung-ho at this species, you're not going to have any chance for success. Pesticide options. We'll talk about it right now. Pesticide options. There's one systemic pesticide that's approved for natural uses, and that is called Safari. But the issue with Safari is that because it moves throughout the plant and will kill insects as they feed on it, and remember thrips have sucking mouth parts, and that's one of the major causes of damage to the plant. The plant cannot be flowering when you apply this systemic insecticide. So what that means is the few times that we did try it, we had to go through manually and clip off all flowers before administering this chemical treatment. And really that's pretty unsustainable, especially depending on the size of the population that you're dealing with. If it's a small plant, you could absolutely keep this up with repeated treatments, but eventually you would probably run into some chemical resistance and have to deal with those issues. There are some contact herbicides. There's some contact insecticides that we were using after we removed plants to treat the vegetative material before transporting it for incineration. So those would be Conserve, which is spinosad, that's the active ingredient, and Tallstar, which is bifenthrin, is the active. So just jumping into the identification of the insect quickly, you can see just how tiny these guys are. This is why we tell folks to look for the damage and not the insect. It's very difficult to find this insect, even sometimes in heavily damaged Nio individuals. So what that brings us to is the main crux of the presentation, which is our future management action. So on Oahu, like I said, we've transitioned from an eradication phase to an active management phase. So that's at the landowner's discretion. That's going to be based on repeated treatments or removal. Again, that's up to the landowner. We do believe, though, that in order to find a successful treatment matrix, you're going to need some experimental plots, and you're going to have to probably work with the HDOA pesticides division just to try and figure out what kind of chemicals to test out and how to set up that kind of treatment experiment. We don't currently have funding for thrips work in 2021. It also is partially because it now falls outside of our invasive species committee grasp, and that it's no longer really incipient to our island. So our main goal right now on Oahu is to try and prevent thrips from expanding to Maui, Nui, and Kauai. Again, you notice I left Hawaii Island off of that, and that's just because these pests are very prevalent on Hawaii. So we do have some broad suggestions, early detection, rapid response suggestions for our neighbor island land managers. The main one is prepare a coordinated response plan. Make it multi-agency, make it island or countywide if you can. We would not have been able to respond with in any significance to this outbreak without the help of our partners, specifically the staff over at the Department of Forestry and Wildlife here on Oahu. Big thanks to them and all of our partners who were doing little NIO delimiting surveys and monitoring known populations. So really, if you are going to make the decision to tackle this pest, should it show up on your island, you really want as many hands on deck and as many tools in the toolkit as possible. You're going to want to delimit NIO populations as both upright and papa. NIO is one of the most prevalent plants used in Oahu landscaping. I'm fairly certain that translates over to the neighbor islands as well. And it's also a common, the sandwichensis is also a common plant that can be found in natural wildland locations. Once you do delimit those populations and know where your plants are, you're going to want to monitor them quarterly. That's what OISC was doing for the populations that we knew about. Unfortunately, when thrips was detected, it was not in one of our locations that we had known. So on the ground kind of management suggestions, we still recommend removing the entire plant. We had previously recommended just removing and bagging damaged foliage. Unfortunately, what we discovered is because these insects are so good at dispersing across the landscape, any kind of disturbance, including going in and cutting off damaged pieces of foliage, but leaving the remaining tree, you're releasing a lot of those insects up into the air and likely aiding their spread. So instead of doing that, we recommend just cutting the whole tree down, the whole plant down, or removing all of the patch of Niopapa at once, rather than trying to meticulously go through and remove only damaged foliage. After you remove that, you want to put it in a big pile and you just want to treat it with Tall Star or Conserve. Those are the two contact insecticides that I mentioned previously. And then you're going to want to tarp the pile of vegetation securely for 6 to 8 weeks on site. The reason we do that is because from start to finish, the life cycle of the thrips should expire just under 6 weeks. So if it's 6 to 8 weeks, you should be safe, although you're going to want to check and see if thrips are still present in your pile before moving on. And if there are still thrips, of course, repeat the treatment and continue tarping. If you are at a site that you cannot tarp, we ran into this several times, we would recommend at that point just putting them into the contractor bags. Usually you have to put them into multiple bags and cut the pieces extremely small. Otherwise, you're going to puncture your bag. And if it's in the back of a pickup truck and you're trying to transport it to an incineration plant or any place of disposal, you're likely aiding in the spread as well. And then, yeah, the best way is incineration. So here on Oahu, we have the H Power plant that incinerates a good portion of our rubbish. So we do have a permit to drop plant material off directly to them. So we were doing that, but you could also, as long as the bags are secure, if you wanted to put it in your rubbish, as long as you know it's going to the incineration plant and not the green waste, that would also be one option. And then decon, right? If you don't consider decon, you're not going to have success. So that's twofold. We want a lot of site decon at these areas. Park your vehicles far away, inspect all your equipment, and then make sure everything is secure before leaving the site. You're also going to want to decontaminate each other, all the folks who are out there helping. You're going to want to change out of clothes at the site. You're going to want to inspect each other, brush off, and then when possible, wear species-specific clothing and Tyvek protection. And that was my whirlwind quick NIO thrips update / recommendation. Thanks to all of our partners. Thanks to all of our funders. And if you have folks have questions, feel free to reach out to us. Our emails are up at the top. You can also visit us at oahuist.org, and you can find us on various social media platforms. I think we're the most active on probably Instagram and Facebook. And that's it.
Thank you, Nate. Very important project and plans to restore our native habitat, native landscape. We need to move a little bit quicker. Moving along, we have a last presentation. Which is the most current one, which was in the news, the recent detection of coffee leaf rust on Hawaii Island and the Maui. And the title of the talk is the Hawaii Department of Agriculture Response to the Discovery of Coffee Leaf Rust in Hawaii by Kevin Hoffman. And Kevin Hoffman is the administrator for the Plant Industry Division, Hawaii Department of Agriculture. Kevin, whenever you are ready.
Okay, thank you, Rashawn. Let me see if I can share my screen here.
Yes, so just one more thing. We might need to extend this meeting by maybe 10 minutes or so, just to see heads up, because I think, yes, we can extend. And if anybody needs to leave, please feel free to leave. We will need to extend by a little bit, I think. Thank you. Kevin.
Okay.
Yeah.
Let me see. Okay, can you see that? You see the?
Yes.
Can you see my screen? Okay.
Yes.
Let's start the slideshow then. Okay. Good afternoon, everybody. As Rashawn said, I'm Kevin Hoffman, Administrator of the Plant Industry Division with the Hawaii Department of Agriculture. So today I'm going to give a brief talk on the department's response to the discovery of coffee leaf rust. And if you're not familiar with coffee leaf rust, it is one of the two pests that are specifically noted as justifying restrictions on the introduction of coffee into the state as mandated by Administrative Rules Section 4-72. There's also a federally regulated pest. It's a devastating coffee pathogen, first discovered in Sri Lanka in 1869. Subsequently, it spread to all the major coffee producing areas in the world, except Hawaii, until its detection here in October. It can cause severe defoliation of coffee plants, resulting in greatly reduced photosynthetic capacity, and depending on its prevalence in a given year, both the vegetative and berry growth can be greatly reduced. There are multiple long-term impacts of CLR, including dieback, resulting in an impact on to the following year's crop. In some places, they've estimated losses ranging from 30 to 80% if the disease is not managed. Initial symptoms of coffee leaf rust are small yellow spots on the upper sides of the leaves, followed by a yellowish-orange powdery rust on the undersides of the leaves. And this powdery stuff are the spores, and the spores can spread readily through physical contact. They're both airborne and waterborne. So here's over to the left are some typical symptoms of coffee leaf rust. Looking at the bottom of the leaves, you can see the yellowish-orange spots. Then a pull up over to the right, we can see like little individual spores. And the impacts on wild coffee. Coffee leaf rust was first found on Maui. These are wild coffee plants. They're essentially been defoliated by the disease. At this point, they're pretty much just sticks in the ground. And we've seen similar effects to that on wild coffee in parts of the Big Island as well. And going back to the chronology, on October 20th, UH Extension Agent Andrea Kawabata notified the department staff on Maui that a resident small grower from the Haiku area of Maui will be bringing by leaf samples which display symptoms of CLR. The next day, the samples arrived, and based on the physical characteristics and host expression, staff tentatively identified the pathogen as coffee leaf rust. Samples were submitted to the UH CTAR for initial identification, and because it is a federally regulated pest, we had to send samples to the United States Department of Agriculture National Identification Services for official identification confirmation. We CTAR on October 26th, tentatively identified the pathogen as CLR, and on 2 days later, USDA NIS notified HDOA that the identification was confirmed as coffee leaf rust. That triggered surveys on Maui, and within a day, we found plants in several locations, including wild coffee, that exhibited symptoms. That triggered surveys on all the islands, and on October 31st, Andrew Kawabata picked up a sample of coffee leaf rust collected by a grower on the west side of the Big Island in the Kona area. The sample was taken to Dr. Lisa Keith at the USDA Agricultural Research Service Pacific Basin Agricultural Research Center, PBAR, that day, which was a Saturday, and then tentatively identified as coffee leaf rust. That evening, sample was submitted to USDA NIS, and the department was notified on November 10th. It was confirmed as coffee leaf rust. Interestingly, 2 samples from the east side of the Big Island, the Hilo area, were also sent to USDA NIS for identification, but the rust was not detected. And so samples will be resubmitted to USDA NIS. A curious thing is that we can find scattered spores in the Hilo area, but the disease does not seem to be expressing itself with classic symptoms. So it's a little bit odd. I'm not sure what's going on there. Anyway, the department is continuing to survey on Maui and the Big Island to determine the extent of the infestations. Surveys are ongoing on Kauai and Oahu, which have been negative for CLR to date. Partners on both Lanai and Molokai have been informed and asked to look for the disease, and no reports of coffee leaf rust have been received. So at this point, it's just only known from Maui and the Big Island. This is a map showing the Maui survey as of the end of November. You can see the red stars are sites were considered positive for the rust, and they're primarily on the north half of the island. The black pins are negatives, spots where they went out and did not find the disease. An interesting thing is that the disease likes moisture, and so some of these positive spots on Maui were in fact negative when we first went through and surveyed them in October. But then as the rains came in November, those areas started to show symptoms because the rain presumably promoted the expression of the symptoms and the growth of the fungus. It's fairly widespread on Maui. The Big Island, positive finds have been all on the west side in the Kona area and down toward the south. As I said, in the Hilo area, we found some spores, but not really much in the way of disease expression. And we've surveyed all the way up to the northern part of the island without finding anything. So it seems to be more limited on the Big Island than it is on Maui as far as its distribution. On Oahu, we've surveyed a number of sites, including commercial grows, and have not found any signs of the disease. So the black pins are the negative survey sites on Oahu. And on Kauai, we've surveyed a number of sites on the east side of the island, and have not south and east sides of the island, and have not found anything. And what we have kind of determined is from where we found the disease being expressed is that there are certain characteristics in the environment that promote the expression of the disease. And first is water, rain, mist, overhead irrigation, splashing water, ways to get water actually physically on the leaves. High humidity seems to promote growth of the fungus. Seems to be a somewhat narrow temperature range where it doesn't like anything much below 59 or much over 85. It does better in partial shade than it does in full sun. And it also does better in areas that are sheltered from the rain. In regulatory action, on November 13th, the Advisory Committee on Plants and Animals voted to recommend approval of an interim rule that restricts the inter-island movement of potentially infested articles from Maui and the Big Island. 4 days later, the department chairperson accepted the recommendation and approved the interim rule with an effective date of November 20th. So just about 1 month after the first detection. So the interim rule. The interim rule prohibits the intrastate movement of coffee plants, including hybrids and plant parts such as green beans, fruits, leaves, stems, twigs, cuttings, wood, logs, mulch or green waste, used coffee-related packing materials such as coffee bags, and any previously used equipment used to harvest, transport, or process coffee plants or plant parts, all of which are considered to be potential carriers of the fungus. From the island of Maui, Big Island, or any other island subsequently confirmed with coffee leaf rust. And this restriction on movement is in place for up to 1 year from the effective date. Now there are some exceptions to the restrictions that are available via permit issued by the department, so I'll go into those next. So there are 6 exceptions. So for green coffee beans for roasting or other non-propagated plant parts, they're shipped to an approved facility located on an uninfested area that is located at least 2 miles from the commercial coffee area or subject to an approved treatment. This would be something for example, shipping green beans from the Big Island to Oahu for roasting. Second one is for plants from department certified nurseries, green coffee beans for roasting or other propagated plant parts. They're transshipped through an uninfested area of the state with a final destination outside the state. And by transshipping, I'm talking about, okay, the material gets put into one conveyance, for example, a barge on the Big Island. It goes over to Honolulu, it's taken from that barge and put onto something else. So it's actually it's transferred from one carrier to another. In that situation, a transshipment permit, along with that specifies the mitigations necessarily would be needed. If something is, for example, put into a container on the Big Island, the container goes over to Honolulu, the material does not leave that container, then it's shipped somewhere else to the mainland, that does not need a permit because it never is not moved from one carrier to another. In other words, it's not moving away from the port environment into an area where it could infect coffee plants. Number 3, for previously used equipment that is designed to harvest, process, or transport coffee plants or plant parts. Number 4, there's an exception for scientific studies or diagnostic uses. There's a permit for moving material between infested islands. So there's some mitigations that need to be taken there as well. You don't want to increase the fungus load on one island, even though it's there in some places. And finally, there's a permit for the limited quantities of coffee plants for propagation that are moved from an infested area to a non-infested area. And of course, these are subject to a 1-year quarantine in a state-run facility, provided that the Board of Agriculture can reduce that quarantine period on a case-by-case basis. The department has engaged in a number of outreach efforts. We notified the coffee industry of detections as they occurred and issued press releases. We posted a field guide with a submission form that the growers and the public can use to kind of identify suspect symptoms and then submit information to us so we can kind of track where the disease is at. We've worked closely with UHCTAR. They produced a sanitation protocol document. CTAR has also produced spraying guidelines for coffee leaf rust suppression. Being an extensible publication that's in 4 languages now. And CTAR is also drafting farm surveying, sampling, and monitoring guidelines. They created a website that has a lot of all this information is kind of gathered in one place. And the industry has been very active in engaging with the department and engaging with their members. And so the webinars have been sponsored by the Hawaii Coffee Association and by the Kona Coffee Farmers Association to educate their members on coffee leaf rust. So it's been a I've been very pleased, very collaborative effort between the department, the university, the industry, and USDA. And this is just a first page of the coffee leaf rust field guide that HDOA put out, showing areas to look for on the bottoms of leaves to indicate potential CLR symptoms. And with that, I'm open for questions.
Thanks, Kevin, for the most recent news and the regulatory protocols on coffee leaf rust. Regarding this, I have a question. So if a coffee grower has a farm at one site and the milling place at the other side, maybe it's more than 2 miles away from the farm, what will be the protocol for that?
Well, if it's uninfested, the interim rule addresses inter-island movement. But it does not, it does not address intra-island movement.
So
we tried that with coffee berry borer and with banana bungee top, and neither worked very well. It's just the way the islands are, it's very difficult to restrict movement of all those kinds of articles from one part of an island to another. We have our best shot at preventing inter-island movement through the regular quarantine inspection processes and so forth.
Thank you, Kevin.
Sure.
Thank you, all the speakers. We have a time, maybe 10 more minutes for the questions for all the speakers. Yes, there is some questions in the chat box. So Chelsea has a question. Any ideas on time and source of introduction? This is for coffee leaf rust.
Coffee leaf rust. Not really. It's been here probably at least a year. It's fairly widespread on Maui. It's in wild coffee. Probably more than a year, be my guess. I can't really, as far as how far back it goes, I'm not really sure. And as far as the source, samples have been sent for genotyping as well as race determination to researchers at Purdue University and then also in Portugal. And so once we get that information back, then we may narrow down the part of the world that our coffee leaf rust came from, or parts of the world, depending. It could have also been multiple introductions.
So maybe I have another, just curious. If the coffee plant is infected with the coffee rust, they show some symptoms as a leaf spot only, but doesn't show rust. Is that I think it's sometimes in the coffee rust disease, yeah?
Yeah,
but it doesn't show the rust.
There's another disease, Cercospora, that occurs that's widespread on the islands that causes the same kind of, more or less the same kind of yellow spotting on leaves. However, it does not produce the spores on the bottom of the leaf. So there are some. We're working with CTAR and USDA ARS to put together a publication to try and show the difference between the two. It can be a little tough depending on how advanced the symptoms are of the coffee leaf rust. They can look very similar. So we are working on that. But yeah, there's something else out there that looks, to the untrained eye, somewhat like coffee leaf rust. It's already there.
Thank you, Kevin.
Sure.
Erin has a question to, maybe, I don't know if Jane can answer this. For parakeets, any plans to monitor the situation on Oahu? Jane?
Unfortunately, I don't know of any formal efforts right now. Occasionally, the USDA will control them there, but nothing too extensive or formal at this point. Certainly something we'd love to do in the future, but I think we all know that funding is going to be in a bit of a crunch for the next year or so. So hopefully soon.
Thanks, Jane. Cookie has a question. This is for JD. So she wants to know about the status of Pecum steam unit treatment for rod.
Yeah, the data from that study are still being written up for submission to the Department of Ag, and so then it would be approved as a treatment method. The unit I showed you was an experimental unit that was shipped here for the purpose of the experiment and went home, but there's one mill on the island that has purchased their own small unit right now. That they should be able to go forward with that.
Thanks, JD. So we have a few more minutes. I think we can spend some time asking questions. If you have questions, please type in the chat box, or you can unmute yourself and ask some questions.
Hi, Roshan. Yeah,
Yes,
hi.
yes.
I have this is Cheng from PEPS. I have a question to the CRB team. Have you guys taken the canine to Ted McLennan golf course recently? I'm asking because I have an ongoing CRB trial over there, and we are seeing actually a lot more new damages over there. And then, as you probably know, they are removing the big compost pile in the center of the golf course. I don't know whether you have any plan or recently took the dogs over there. It's just, my graduate student and me, we couldn't figure out where the breeding site would be, but there must be some breeding sites nearby the property, nearby the golf course, or in the golf course itself. It's just, hard to figure out. Yeah.
Yeah, definitely. I'm not sure the date, but definitely within the past even few weeks to maybe a month ago, our canines have been out, and similarly, with the removal of that big pile, we're following up with our contractors about some material that still remained at the site, and they're looking into some other options for treatment there too, whether that's some more pesticide injection work, or if there's options to treat with the granular pesticide. But yeah, thank you for pointing that out. Definitely a conversation that we're hoping to pursue. Kylie, anything to add to?
No, that was great. Yeah, we're still working with Ted McLennan Golf Course on what the next step for that area is going to be.
Okay, thank you. Just a quick comment. I'm happy to see that my picture was included in your presentation. I think Rochelle was there when we were doing the injection, right? Back in 16, I believe. Yeah.
Yes. Thank you. Thank you, Kylie and Cookie. Any more questions? So yeah, I just wanted to share also Plant Industry Division SGOA Administrator Kevin Hoffman is retiring soon, retiring at the end of this year. So hopefully he will be around for another two weeks, I think. So congratulations and happy retirement for Kevin Hoffman.
Well, thank you, Roshan. Yeah. Yes, it's been a pleasure working with a lot of you on a lot of different projects over the past couple of years. So, I wish you the best going forward, and have some great holidays.
Thank you.
Yep.
You have a happy retirement.
Thank you.
So, any last minute questions?